welcome back to
The GD Podcast!
So for today's podcast nga, meron tayong viewers na nag-ask na as a first-year nursing student daw, what is the Krebs Cycle? So Resha, can you define the Krebs Cycle? Ano nga ba yung role nito sa cellular respiration?
The Krebs cycle, also known as citric acid cycle, is a series of chemical reactions used by all aerobic organisms to generate energy. It plays a crucial role in cellular respiration by oxidizing acetyl-CoA to produce ATP, NADH, at saka yung FADH2. Ito ay essential for electron transport chain.
So, yung location na to within the cell is after you eat, your body break down food into a molecule called acetyl-CoA. So, the Krebs cycle take this molecule and process it through a series of steps in the mitochondria of your cells. So, after natin ma-define yung Krebs cycle, we're going now to identify the reactants and product. Anton, what are the reactants?
Okay, let's explore the step-by-step process of the Krebs cycle. In the early steps of the Krebs cycle, the citric synthase or formation of citrates links to the oxaloacetate substrate, which can then bind to acetyl-CoA's acetyl group, which then releases the coenzyme A.
And to further explain, this produces the very familiar and common citric acid called a citrate. It is this 6-carbon molecule that will be broken down and remolded back into oxaloacetate.
Unfortunately, citrate is too stable for the reactions that follow. So in the second step, which is the formation of citrate isomers, the aconitase links to the citrate to move one of its oxygen atoms to create a more unstable citrate isomer.
It does this by extracting a water molecule, producing a cis-aconitate, and then reattaching the water to produce D-isocitrate.
With the citrate rearranged, the process begins by decarboxylating and oxidating the isocitrates. The isocitrate dehydrogenase links to the D-isocitrate, which transfers an electron to the nicotinamide adenine dinucleotide, producing its energized form NADH.
With the electron removed from NADH, the enzyme then detaches a carbon atom to form a molecule of carbon dioxide. This transforms the substrate from a 6-carbon molecule to a 5-carbon molecule.
This step involves a highly developed complex of 24 enzymes. Labeled here are alpha-ketoglutarate dehydrogenase. This complex also transfers electrons to NAD producing NADH, removes another carbon atom as carbon dioxide, and relinks the coenzyme A to the substrate, resulting to the formation of succinyl-CoA. In this step, succinyl-CoA is formed. This step directly produces ATP because the substrate's link to coenzyme A is sufficiently energetic to power the reaction.
In the mitochondria, the enzyme links to the succinyl-CoA and uses the energy from releasing the coenzyme to add a phosphate to GDP to produce GTP. In the cytoplasm, a variation on this enzyme can produce ATP directly. This also begins the manipulation of the substrate to reproduce its original form.
With the carbons removed, the rearrangement process begins manipulating the hydrogen. When the succinate dehydrogenase links to the substrate,
It releases 2 hydrogen atoms, attaching them to a carrier, ubiquinone, or flavin adenine dinucleotide. With the additional 2 electrons, ubiquinone forms ubiquinol, which is then transferred to power the electron transport chain, resulting to the formation of fumarate.
to form malate, fumarase continues the rearrangement process by adding hydrogen and oxygen back into the substrate that had been previously removed. Finally, the malate dehydrogenase recreates the oxaloacetate substrate and moves electrons from the NAD to form NADH, the last energy produced by Krebs cycle. Interestingly, this malate oxaloacetate reaction is also used to move anaerobic energy from the cytoplasm into the mitochondria.
While anaerobic reactions produce NADH, it cannot move from the cytoplasm to the mitochondria to be processed in the electron transport chain. But the malate can be transported across the mitochondria's membrane, so the anaerobic NADH transforms oxaloacetate into malate, which is then converted back into oxaloacetate to produce NADH for the production of ATP.
Reviewing the whole process, the Krebs cycle primarily transforms the acetyl group and water into carbon dioxide and energized forms of the other reactants.
Additionally, many of the enzymes and substrates are also intermediaries in other biochemical reactions of both amino acids and fatty acids. As a result, the Krebs cycle is the primary method of cellular energy reproduction.
So after natin ma-identify ang reactants and product, we're going now to explore the step. Joseph, can you describe the individual reactions na na-occur during the cycle?
Okay, we can start by citric acid. So, citric acid cycle begins with acetyl-CoA, combines with oxaloacetate to form citrate, which then undergoes a series of transformations. Citrate is rearranged into isotrate, and through oxidation, it releases carbon dioxide and produces NADH. Then a ketoglutarate is formed and further oxidized, releasing another CO2 and generating more NADH. As the cycle continues, as succinyl-CoA turns into succinate, producing ATP, which is the direct source of energy for the cell. So finally, succinate is converted into fumarate, which becomes malate. The cycle finishes as malate and is oxidized back into the oxaloacetate, creating one last NADH. Overall, each cycle generates 3 NADH, 1 5H2, and 1 ATP, which are essential for producing energy in cells through the electron transport chain.
Now na alam na natin yung steps. We're going now to discuss the importance of ATP. Bianca, can you discuss the importance of ATP?
So, ATP or adenosine triphosphate is commonly known as the energy currency of the cells. So now we will delve more why ATP is called as the energy currency of the cell. Let us put ATP into simpler terms in which let's imagine that our body is just like a big city. It needs electricity in which in our cells, ATP is the electricity. This ATP provides energy needed for many cellular processes. Every time we move our muscles, blink our eyes, we spend ATP in our body. But first, let us know where does ATP comes from. Let us again imagine that our cells as little factories. When we eat food, it's like bringing raw materials to the factory. It is what we call the Krebs cycle. The Krebs cycle is one of the production lines inside that factory, turning those raw materials into usable energy. But how exactly does it do that? Just like what they said earlier, Krebs cycle, also known as citric acid cycle, comes in. It is a process in our cells that helps produce energy. It is a series of chemical reactions that converts food into energy. This cycle starts with acetyl-CoA, which comes from carbohydrates, fats, and proteins. This enzyme is combined with oxaloacetate to form citrate that undergoes series of transformations. Throughout the cycle, the breakdown of acetyl-CoA results in the production of electron carriers, which are the NADH and FADH2. They store energy that will be used later in ETC or the electron transport chain. So every turn of Krebs cycle, there is a small amount of ATP that is directly produced. Significantly, NADH and FADH2 carry electrons to electron transport chain where much larger amount of ATP is generated. This ATP provides the energy needed for various cellular activities, making it fundamental to the functioning of all living organisms. So to sum it up, ATP is like the currency your cells spend to get things done. So, and the Krebs cycle is one of the main ways your body produces this currency.
Ngayon na na-discuss na ni Bianca yung importance ng ATP, Santino, can you label each step and molecule sa loob ng Krebs cycle?
So here's a step-by-step breakdown of the cycle. So the first one is the citrate formation or the acetyl-CoA, a 2-carbon molecule combines with oxaloacetate, a 4-carbon molecule to form citrate, a 6-carbon molecule. This reaction is catalyzed by the enzyme citrate synthase. So the second is isocitrate formation. Citrate is converted to its isomer isocitrate. This reaction is catalyzed by the enzyme aconitase. So the third is alpha-ketoglutarate formation. Isocitrate undergoes oxidative decarbosylation, releasing a carbon dioxide molecule and forming a alpha-ketoglutarate, a 5-carbon molecule. This reaction is catalyzed by the enzyme isocitrate dehydrogenase, which also produces NADH, a reduced coenzyme. The fourth one is succinyl-CoA formation. An alpha ketoglutarate undergoes another oxidative decarboxylation, releasing a second carbon dioxide molecule and forming succinyl-CoA, a 4-carbon molecule. This reaction is catalyzed by the enzyme alpha ketoglutarate dehydrogenase, which also produces NADH. So the fifth one is succinate formation. Succinyl-CoA is converted to succinate, releasing coenzymes A-CoA. This reaction is catalyzed by the enzyme succinyl-CoA synthetase, which also produces either ATP or GTP depending on the organism. The sixth part is fumarate formation. Succinate is oxidized to fumarate, producing FADH2, reduced coenzymes. This reaction is catalyzed by the enzyme succinate dehydrogenase. The seventh is malate formation. Fumarate is hydrated to form malate. This reaction is catalyzed by the enzyme fumarase. The eighth is oxaloacetate regeneration. Malate is oxidized to oxaloacetate, producing NADH and regenerating the starting molecule of the cycle. This reaction is catalyzed by the enzyme malate dehydrogenase. Overall, each turn of Krebs cycle produces 2 carbon dioxide molecules, 1 ATP or GTP molecule, 3 NADH molecule, and 1 FADH2 molecule. These products are then used in the electron transport chain to produce ATP, the primary energy currency of cells. So that's the process and step by step of this Krebs cycle.
Ngayon na na-discuss na Santino yung diagram ng Krebs cycle, we're now going to explore the medical implication. Jeff and Joshua, can you guys explain how disorder or diseases can affect the Krebs cycle, and can you guys also discuss the potential treatments or the therapies?
So, partner Joshua, marami tayong disorders and disease that can affect the citric acid cycle or the Krebs cycle. So, the first one, we have the pyruvate dehydrogenase deficiency. So, it affects the conversion of pyruvate to acetyl-CoA. So, kapag hindi na-convert yung pyruvate into acetyl-CoA, naiipon yung pyruvate and it convert to lactate that can lead to lactic acidosis. So,
Ayan na nga, tuloy mo lang partner, tuloy mo lang.
So, lactic acidosis, partner, is one of the symptoms, while the one is the neurological issues. So, the treatment for that is dietary management or high-fat and low-carbohydrate diet. So, nandito rin si thiamine supplementation, which can help to turn food into energy. It helps our nervous system to be healthy. So, the second one, partner, is the succinyl-CoA synthetase deficiency. So, it affects the conversion of succinyl-CoA to succinate. So, the symptom of this is muscle weakness o yung panghihina ng kalamnan natin. And the energy deficiency o yung kakulangan sa energy. So, yung treatment is supportive care and potential dietary medication. So, what will you mayambag mo?
Okay. So, ituloy ko lang yung isa yung mga sa mga disorder o diseases. So, isa sa mga to yung mutation of the isocitrate dehydrogenase. Isa tong disease or disorder related to the Krebs cycle, for example, would be the mutation of the isocitrate dehydrogenase, which is found in several cancers. Now, instead of the alpha ketoglutarate, which is what happens when isocitrate is oxygenated within the Krebs cycle, a mutant IDH enzyme would instead create 2-hydroxyglutarate, which causes DNA and histone hypomethylation, leading to neoplasia. Now, there are mutants IDH1 and IDH2. A mutated IDH1 has a treatment called an IDH1 inhibitor or ivosidenib. ivosidenib. Yeah. While for the IDH2, it's called enasidenib or Inhifa. As for the second, I'm just gonna catch it. As for the second disease that I will state, it's the fumarase deficiency. It's a problem in the FH gene with that you will not produce enough of the fumarase enzyme, which is important to convert fumarate to marate. With the deficiency of the enzyme fumarase, genetic problems may occur. Pero ang problema kasi doon, ito naman sa pangalawang disease, wala siyang treatment. Ayun. So treatment may be offered such as medications to reduce seizure, and a feeding tube may also be inserted to make sure that your body is receiving the correct nutrients.
Ngayon nga na alam na natin yung medical implication from Jeff and Joshua. Ngayon naman, Isa, ano ba yung application ng Krebs Cycle sa industrial uses natin?
We all know that the Krebs cycle or citric acid cycle ultimately allows the food we eat, such as glucose, to be converted to energy. But it also plays a significant role in various industries, particularly in food biofuel production. So now, how does the Krebs cycle contribute to food production? The Krebs cycle facilitates cellular respiration in plants, animals, and microorganisms. In energy production, the cycle generates ATP, the energy currency of the cell, enabling metabolic processes necessary for growth and development. In metabolite synthesis, it produces key intermediates that serve as the building blocks for amino acids, fatty acids, and other biomolecules essential for plants and animal growth. In plants, the Krebs cycle works in conjunction with photosynthesis, utilizing products from light reactions to fuel the cycle, leading to the synthesis of carbohydrates. In food production naman, microorganisms use the Krebs cycle during fermentation, producing compounds like lactic acid and ethanol, which are important in making the bread and alcoholic beverage. Making the Krebs cycle necessary for energy production and synthesis of vital nutrients in the food supply chain. Now, how does the Krebs cycle contributes to biofuel production naman? In our body, the Krebs cycle produces sufficient amount of ATP for other cycle in our body, and this ATP to continue the cycle happening in our body and complete the cellular function. The Krebs cycle is crucial in biofuel production, where organisms or algae are often utilized to ferment biomass, generating intermediates that can enter the Krebs cycle. This process helps maximize energy extraction from organic materials, ultimately contributing to the production of biofuels like ethanol and biodiesel. Optimizing these pathways can enhance yield and efficiency in biofuel processes.
So that's it for the Krebs Cycle. See you guys sa next podcast namin. Kung meron kayong mga question, pwede nyong i-send yan sa ggpodcast@gmail.com or sa aming Facebook page na GG Podcast. Ililink namin. Yun lang, and see you guys! Bye!
Increase in heart rate, increase in heart rate, increase in heart rate, increase in heart rate.